What is culture media? The media is a source of nutrients to support the growth of the microorganisms in vitro. The media helps in the growth and counting of microbial cells, selection of microorganisms, and survival of microorganisms. The culture medium can be liquid or gel. Common ingredients of culture media: peptone, source of carbon and nitrogen; beef extract, source of amino acids, vitamins, minerals; yeast extract, source of vitamins, carbon, nitrogen; distilled water; agar, solidifying agent. How to prepare culture media: weigh the amount of ingredients powder on weighing machine, dissolve the ingredients in distilled water, adjust pH of the medium if needed, add agar and boiled it to dissolve, pour the media into flask, autoclave the media when ingredients fully dissolve. Sterilization is done in autoclave to prevent from contamination, at 121 OC for 15 min at 15 lbs. After the autoclave, place the media flask in laminar airflow. Sterilize the laminar airflow with 70% alcohol. A bit cools down the media and pours into sterile Petri plates for solidification. Then sample is ready to spread, spreader, germ streak, inoculation loop, on the medium for identification or isolation of microbes. Seal the Petri plates with paraffin, label them. Keep them inverted in incubator at 37 OC for 24 HRs. Observe the result next day. Colonies formation is visible on the media. What is a defined medium? A defined medium has a known quantity of all ingredients like carbon source, glucose or glycerol, and nitrogen source, ammonium salt or nitrate as inorganic nitrogen. The medium needs in metabolic, nutritional, and physiological growth experiments. Chapek, Docs, medium. What is an undefined medium? This medium has different complex ingredients in unknown quantities, for example, yeast extract, beef, various salts, and enzymatic protein. Potato dextrose, agar. Makonki, agar. What is complex media? This media is other than basal media. It has added ingredients to bring the characteristics of microorganisms with unique nutrients. Types of culture media. Types of culture media based on consistency, physical state. 1. Solid medium. 2. Semi-solid medium. 3. Liquid medium. 1. Solid media. Principle of solid media. It is for the isolation of bacteria as a pure culture on a solid medium. Robert Koch realized the use of solid media. Agar is used to hardening the media at 1.5–2.0% concentration. Solid media allows the growth of bacteria as colonies by streaking on the medium. It's solidified at 37 degrees Celsius. Agar is an unbranched polysaccharide extracted from red algae species like Gelidium. Colonies identification is done on this medium. Examples of solid media: nutrient agar, Makonki agar, blood agar, chocolate agar. Growth of bacteria on solid medium appear as smooth, rough, mucoid, round, irregular, filamentous, punctiform. 2. Semi-solid media. Principle of semi-solid media. This media shows the motility of bacteria and the cultivation of microaerophilic bacteria. This media has agar at a concentration of 0.5% or less. It has a jelly consistency. Examples of semi-solid media: Stewart's and Amy's media, Hugh and Leifson's oxidation fermentation medium, and mannitol motility media. The growth of bacteria in semi-solid appears as a thick line in the medium. 3. Liquid media. Principle of liquid media. This media shows the growth of a large number of bacteria. It is called broth that allows bacteria to grow uniformly with turbidity. The growth occurs at 37 OC in an incubator for 24 hours. Liquid media don't have the addition of agar. It is for fermentation studies. Examples of liquid media: nutrient broth, tryptic soy broth, MRVP broth, phenol red carbohydrate broth. Growth of bacteria in liquid media. Turbidity is seen at the end of the broth. Types of culture media based on chemical composition, application. There are 7 routine laboratory media: basal media, enriched media, selective media, enrichment media, indicator media or differential media, transport media, storage media. 1. Basal media. This media is simple as it enhances the growth of many microorganisms. It's a routinely used medium in the lab, having carbon and nitrogen. This media allows the growth of non-fastidious bacteria without any enrichment source, used for subculturing. It's a non-selective medium. Staphylococcus and Enterobacteriaceae grow in this media. Examples of basal media: nutrient agar, peptone water. 2. Enriched media. This media requires the addition of other substances like blood, egg, or serum. An enriched media allows the growth of devised microorganisms but inhibits other and fastidious microbes grow, as they require nutrients like vitamins and growth-promoting substances. Example of enriched media: blood agar, chocolate agar, LSS, Monsor's taurocholate, Lowenstein-Jensen media. Blood agar identifies hemolytic bacteria, chocolate media for N-gonorrhea. 3. Selective media. As by name we can tell, this media shows the growth of selective microbes or desired microorganisms and inhibits the growth of unwanted microbes. The inhibition occurs by adding bile salts, antibiotics, dyes, pH adjustments. Media is agar-based. Any media is possible to transform into selective by adding inhibitory agar. Examples of selective media: SN, media bacteria 1, mannitol agar. It has 7% of sodium chloride that inhibits the growth of other microbes and promotes the growth of Staphylococcus. It has phenol red dye that produces acid. Staphylococcus used the mannitol for the acid production, and the color of phenol red changes from red to yellow. Selective for Staphylococcus aureus 2. Salmonella, Shigella. Agar, deoxycholate agar. It is used for the isolation of Salmonella bacteria that causes typhoid. Selective for Shigella. 3. McConkey agar. It has bile salts that inhibit the growth of gram-positive bacteria. Selective isolation for Enterobacteriaceae 4. TCBS agar, light green translucent media bile salt, inhibits the growth of unwanted bacteria. Selective for Vibrio cholera. V. cholera produces acid by fermentation of sucrose that acts as indicator called bromethymol. Blue and yellow colonies appears. 5. Lowenstein-Jensen media. It is made selective by adding malachite green and stops the unwanted growth of pathogens. Selective for M. tuberculosis. 4. Enrichment media. It is a liquid medium, which also permits the growth of desired bacteria at a low density. The media provides an environment and conditions as selective media and inhibits unwanted bacteria from growing. It is for the isolation of the soil and fecal microorganisms. Examples of enrichment media: selenite F broth does the isolation of Salmonella typhi from a fecal sample. Selenium allows the growth of desired organisms and detection levels increase for intestinal flora. 5. Indicator or differential media. This media shows visible changes due to the presence of an indicator. It differentiates bacteria based on colony color growing on the same plate. Biochemical characteristics show organisms' growth, with chemical indicators like neutral red, phenol red, methylene blue. Examples of indicator or differential media: mannitol salt agar. Mannitol fermentation shows yellow color colonies. Blood agar is used to differentiate between hemolytic and non-hemolytic. McConkey agar produces pink colonies due to lactose utilization, and non-lactose shows pale color colonies. 6. Transport media. The media transports specimens after collection to control the overgrowth of organisms. For the cultivation, this media acts as temporary storage. It also maintains the viability of pathogens in the specimen and prevents them from drying. Examples of transport media: Stewart's transport medium, lacks carbon, nitrogen, growth factors. Carey Blair's transport media and VR are used to transport feces samples from cholera patients. Pike's medium helps to transport streptococci from throat patients. 7. Storage media. It maintains the longevity of bacterial culture. Examples are cooked meat broth, Na egg saline. Types of culture media based on oxygen requirement. Microorganisms have different requirements for growth depending on oxygen requirements. 1. Aerobic media. In this media, it is easy to cultivate microbes. On solid media, the growth occurs by keeping the culture in the incubator. It shows the growth of non-fastidious microorganisms. Examples of aerobic media are liquid media, solid media, peptone water, 1% peptone plus 0.5%, NaCl plus 100 mmol water, nutrient agar, nutrient broth, plus 2% agar. 2. Anaerobic media. The media cultivates anaerobic bacteria at low oxygen, reducing oxidation reduction potential. Anaerobic media contains extra nutrients like vitamin K, hemin, and oxygen that get reduced by a physical or chemical process. The addition of glucose, 1%. Thioglycolate, 0.1%. Ascorbic acid, 0.1%. Cysteine, 0.05%, or iron fillings added to cause the medium to reduce. The medium is boiled in a water bath to force out dissolved oxygen and packed with sterile paraffin. Examples of anaerobic media RCM, Robertson cooked meat, isolation for Clostridium sp. Thioglycolate broth, it has sodium glycolate that maintains low oxygen. Types of special purpose culture media. 1. Assay media. The media assay vitamins, amino acids, and antibiotics. Example: antibiotic sensitivity test. The media used is Muller-Hinton. Agar has 1.7% agar for better diffusion of antibiotics. It also contains starch, which absorbs toxins released by bacteria. In this media plate, zone of inhibition is seen around antibiotics. 2. Minimal media. Principle of minimal media. Minimal media is a defined medium with different compositions depending on microorganisms cultured. It contains a carbon source like sugar succinate and inorganic salts like magnesium, nitrogen, sulfur, phosphorus. Carbon is a source of energy. Magnesium and ammonium salts are the sources of ions for metabolism stimulation. Phosphate is a buffering agent, the growth comparison of microbe culture and mutant forms. Minimal media and supplementary minimal media allow the differentiation of wild-type and mutant cells. Use the selection of recombinants for the growth of wild-type microorganisms. 3. Fermentation media. The media is for optimum microorganisms. Fermentation media produce high yields of the product. Media provide energy and nutrients for growth, and medium gives the substrate for the synthesis of products in the fermentation. Fermentation media contains major and minor components. Major components: carbon and nitrogen for energy. Minor components: this contains inorganic salts, growth factors, vitamins, buffer, anti-foaming agents, dissolved oxygen, gases, growth inhibitors, enzymes. The nutrients in fermentation media depend on the organism and type of fermentation process. 2 uses. Growth media. It has low nutrients and creates raw material for further fermentation. Fermentation media. It has high nutrients and creates end products. Example: the yeast requires 1% carbon, but the fermentation of alcohol demands 12 TOS and 13% carbon in the medium. Role of fermentation media. The media has a high level of nutrients, microorganisms, and optimum conditions. During the incubation period under optimum conditions, microorganisms undergo metabolism. Fermentation organisms become hyperactive due to nutrients being in high quantities, and the result is nutrients getting consumed, media partially degraded. The waste effluent is the output product. The death of cells occurs if substrate level reaches the inhibitory concentration and excess substrate causes them to inhibit vital enzymes. Excess substrate increases osmotic pressure and disturbs enzymatic activity in cells. Microbes release excess substrate as partially digested fermentation media and convert it into the insoluble inert compound as reserve food, which is harmless to cells. Example: yeast extract, beef extract, YPD, BMGY. Resuscitation culture media. The resuscitation method is for the stressed bacterial recovery. This is a specialized medium that allows the growth of microbes that have lost the ability to produce because of the environmental harness. The culture provides nutrients and recovers their metabolism. For example, bacteria require histamine for growth and the medium lacks this component. Then it inhibits growth. The same bacterium is put in a medium having histamine, then it starts to grow again, and this medium acts as resuscitation media. For example, tryptic soy agar. Application of culture media to culture microbes, to identify the cause of infection, to identify characteristics of microorganisms, to isolate pure culture, to store the culture stock, to observe biochemical reactions, to test microbial contamination in any sample, to check antimicrobial agents and preservatives effect, to observe microbe colony type, its color, shape, cause, to differentiate between different colonies, to create antigens for laboratory use, to estimate viable count, to test antibiotic sensitivity. Limitations of culture media, risk of cross-contamination, high skill required for optimal results. Increased drying out of media can occur. List of culture media used in microbiology with their uses. A7 and A8 agars. A7 and A8 agars are selective and differential media used for the cultivation, identification, and differentiation of Ureaplasma spp. and Mycoplasma hominis. Alkaline peptone water. Alkaline peptone water is an enrichment broth used for the isolation of small numbers of Vibrio and Aeromonas organisms from stool specimens. American Trudeau Society medium. American Trudeau Society medium is a non-selective enriched medium used for the isolation and cultivation of mycobacteria. Amy's transport medium with and without charcoal. Amy's transport medium is a modification of Stewart's medium. Amy's medium with charcoal is preferred for the isolation of Neisseria spp. Anaerobic blood, AGAR, CDC. The Centers for Disease Control and Prevention, CDC, formulation of anaerobic blood agar is a general-purpose medium used for the isolation and cultivation of anaerobic bacteria. 10B arginine broth. Shepherd's broth. 10B arginine broth is a medium used for the transport and growth of M. hominis and Ureaplasma ureolyticum. Ashdown agar. Ashdown agar is a selective and differential medium for the isolation of Pseudomonas, Pseudomalae. Bacillus cereus, medium B. Cereus medium is an enriched medium used for the isolation of B. cereus. Bactec 12B radiometric medium. Bactec 12B medium, BD Biosciences, is a liquid non-selective medium used for the isolation and identification of Mycobacterium species in conjunction with the Bactec system. Bacteroides bile. Escalin, agar. Bacteroides bile. Escalin, agar is an enriched, selective and differential medium used for the isolation and presumptive identification of members of the Bacteroides fragilis group and Bilophila wadsworthia. Baird-Parker agar base. Baird-Parker agar base is an enriched, selective and differential agar used for the detection of coagulase-positive Staphylococcus S. aureus from food and other non-clinical sources. Barber-Stoner-Kelly medium, Sigma Aldrich Co., St. Louis Mo. Barber-Stoner-Kelly medium is a complicated medium for the isolation of Borrelia burgdorferi. Bile esculin, agar. Bile esculin agar is a selective and differential medium used for the isolation and differentiation of Enterococcus and Streptococcus bovis, group D Streptococcus, from non-group D Streptococcus. Bile esculin agar plus vancomycin at 6 mg/ml. Bile esculin agar plus vancomycin is a selective and differential medium used to identify vancomycin-resistant Streptococcus and Enterococcus. Bile esculin, azide agar, and broth, Enterococcosyl. Bile esculin azide agar, or broth, is a selective and differential medium for S. bovis, group D Streptococcus, and Enterococcus. Bismuth sulfite agar. Bismuth sulfite agar is a highly selective and differential medium used for the isolation of Salmonella enterica serovar typhi and other enteric bacilli. Blood agar. It is the same as Columbia agar with 5% sheep blood. Blood culture media. All blood culture medium formulations are based on a nutrient peptone broth, with variations due to hydrolysis or digestion of the source protein. Bordet Gengu medium. Bordet Gengu medium is an enriched medium used for the isolation and cultivation of Bordetella pertussis from clinical specimens. Brain-heart infusion. Agar. Brain-heart infusion agar is a general-purpose medium used for the isolation of a wide variety of pathogens, including yeasts, molds, and bacteria. Brain-heart infusion agar with 7% horse blood, and brain-heart infusion agar with 1% serum. Brain-heart infusion agar with horse blood or serum enriches the medium for isolation of Helicobacter spp. Brain-heart infusion broth. Brain-heart infusion broth is a general-purpose clear liquid medium that is used to cultivate a wide variety of organisms. Formulations with 6.5% NaCl are used for the isolation of salt-tolerant Streptococcus. Formulations with 0.1% agar that reduce O2 tension favor anaerobes, and formulations with Fildes enrichment are used for the isolation of fastidious organisms such as Haemophilus and Neisseria. Brain-heart infusion vancomycin agar. Brain-heart infusion vancomycin agar is a selective medium used for the isolation of vancomycin-resistant Enterococcus. The base is brain-heart infusion agar. Vancomycin, 6 g/ml, is added to select for vancomycin-resistant Enterococcus. Brilliant green agar. Brilliant green agar is a highly selective and differential medium used for the isolation of Salmonella species except for Serovar typhi. Brucella agar. Brucella agar is a medium designed originally for the purpose of isolating Brucella spp. from dairy products. Brucella agar with cefoxitin and cycloserine. Brucella agar with cefoxitin and cycloserine is a selective and differential sheep blood medium used for the isolation of Clostridium difficile. Brucella agar is the nutritive base. Brucella agar with hemin and vitamin K. Brucella agar with hemin and vitamin K is a general-purpose, non-selective and enriched medium used for the isolation and cultivation of anaerobic bacteria. Brucella agar with 5% horse blood. Horse blood enriches Brucella agar for fastidious organisms, such as H. pylori, by providing both hemin, factor X, and NAD, factor V factors. Brucella broth. Brucella broth is a liquid medium that is used to cultivate Campylobacter species and to identify the organisms to the species level. Buffered charcoal yeast extract, BCYE. BCYE is a specialized enriched agar medium used for the isolation and cultivation of Legionella species from environmental and clinical specimens. Buffered glycerol saline. Buffered glycerol saline is a multi-purpose transport medium. The transport medium has been used for the isolation of bacteria, such as Aeromonas spp, as well as viruses. In addition, glycerol-containing media may also be used for long-term storage of isolates and for transport and storage of biopsy specimens. Burkholderia cepacia selective agar B. Cepacia selective agar is an enriched and selective medium used for the isolation of B. Cepacia. Campylobacter blood agar. Campylobacter blood agar is an enriched selective blood agar medium used for the isolation of Campylobacter species. Campylobacter charcoal differential CCD agar. CCD agar is a blood-free selective medium used for the isolation of Campylobacter from stool specimens. Campylobacter thioglycolate medium. Campylobacter thioglycolate medium is a selective holding medium used for the isolation of Campylobacter species. Carey-Blair transport medium. Carey-Blair transport medium was specifically designed to enhance the survival of enteric bacterial pathogens. Cephaloparazone vancomycin amphotericin B. CVA. Medium. CVA medium is a selective and enriched blood agar medium used for the isolation of Campylobacter species. Cephsalodin ergasin novobiosin CEN medium. Yersinia selective agar, CIN, or Yersinia selective agar, is a selective and differential medium used for the isolation and differentiation of Yersinia enterocollitica from clinical specimens and food sources. Cetramide agar. Cetramide agar is a selective and differential medium used for the identification of Pseudomonas aeruginosa. Charcoal selective medium. Charcoal selective medium is an enriched selective medium used for the isolation of Campylobacter species. Chocolate agar. Chocolate agar is a general-purpose medium used for the isolation and detection of a wide variety of microorganisms, including fastidious species such as Neisseria and Haemophilus. Chopped meat. Glucose broth. Chopped meat glucose broth is an enriched medium that supports the growth of most anaerobes. It is most commonly used to isolate Clostridium botulinum from mixed bacterial growth. Chromagar, Rambach agar. Chromagar is a microorganism-specific chromogenic culture medium used for isolation and identification for a variety of organisms. Chromagar was developed by Alain Rambach, who first formulated agars that were monochromogenic for the detection of E. coli and Salmonella spp. Second-generation agars are multicolor. Both types are differential and selective. The nutritive agar base includes peptone and glucose. Different additives, proprietary chromogenic mixtures and antibiotics have resulted in a series of media specific for such organisms as Listeria, S. aureus, methicillin-resistant S. aureus, MRSA, E. coli O157, yeasts, and other organisms. Columbia agar with 5% sheep blood. Columbia agar with 5% sheep blood is a general-purpose medium used for the isolation of a variety of microorganisms, including fastidious organisms. The addition of 20 g-milliliters of ampicillin per ml is helpful in isolation of Aeromonas. Columbia broth. Columbia broth is a general-purpose clear liquid medium used especially for blood culture medium. The broth supports the growth of a wide range of microorganisms. Columbia colistin nalidixic acid agar with 5% sheep blood. Columbia colistin nalidixic acid agar with 5% sheep blood is a selective and differential medium commonly used in the isolation of gram-positive aerobic and anaerobic organisms from mixed clinical specimens. Cycloserine C-foxidine fructose agar. Cycloserine C-foxidine fructose agar is a selective and differential agar medium used for the isolation of C. difficile. Cysteine albumin broth with 20% glycerol. Cysteine albumin broth with 20% glycerol is used for the transport and storage of gastric biopsy specimens for the recovery of H. pylori. Cysteine glucose blood, agar. Cysteine glucose blood, agar is an enriched medium used for the isolation of Francisella spp. Cysteine tellurite blood, agar. Cysteine tellurite blood, agar, is a modification of Tinsdale agar, and is both a selective and differential medium used for the detection of C. diphtheriae. Cysteine tryptic agar. Cysteine tryptic agar is a pancreatic digest of the casein-enriched medium that is most commonly used for the cultivation and maintenance of fastidious organisms. The addition of 1% carbohydrates and phenol red for the determination of fermentation reactions is especially helpful for differentiating Neisseria spp. Diagnostic sensitivity agar. Diagnostic sensitivity agar is a medium used in the cultivation of organisms for susceptibility testing. DNA toluidine blue, agar. DNA toluidine blue, agar, is a differential medium used most commonly for the detection and differentiation of Staphylococcus spp. Dubose, tween albumin broth. Dubose, tween albumin broth, is a non-selective medium used for the isolation and cultivation of mycobacteria. Egg yolk agar, modified Meklung-Toabe agar. Egg yolk agar medium, modified Meklung-Toabe agar, is a selective and differential medium used for the isolation and differentiation of Clostridium spp. Ellinghausen-McCulloch, Johnson, Harris medium. Ellinghausen-McCulloch, Johnson, Harris medium is an enriched semi-solid medium used for the isolation and cultivation of Leptospira. Enterococcocell agar, it is similar to bile escalinazide agar and broth. Eosin methylene blue, EMB. Agar EMB. Agar is a selective and differential medium used for the isolation and differentiation of enteric pathogens from contaminated clinical specimens. ESP culture system 2. ESP myco medium is used with ESP culture system 2, TREC, diagnostic systems, for the detection of mycobacterial growth. Egg tellurite glycine pyruvate agar, ETGPA. It is similar to the Baird-Parker agar base. Fastidious anaerobic agar, Fusobacterium selective agar. Fastidious anaerobic agar is an enriched sheep blood medium used for the isolation and cultivation of anaerobic organisms. Fletcher's medium. Fletcher's medium is an enriched semi-solid medium used for the isolation and growth of Leptospira. Flextrans viral and chlamydia transport medium. Flextrans viral and chlamydia transport medium, Trinity Biotech, is intended to be used as a transport medium for viruses and/or chlamydiae. GC agar base. GC agar base is a chocolate agar base used with various additives for the purpose of isolating Neisseria gonorrhoeae and other fastidious organisms, such as Haemophilus spp, including Haemophilus ducreyi, and for susceptibility testing of N. gonorrhoeae. GN broth. GN broth is an enriched selective broth medium used for the isolation of gram-negative rods. Specifically, Salmonella and Shigella are isolated more effectively in GN broth than on solid medium alone. Haemophilus test medium, HTM, and broth HTM is an enriched medium used for susceptibility testing of Haemophilus species. Heart infusion agar and broth. Heart infusion agar and broth are general-purpose media used for the isolation of a variety of microorganisms. Incorporation with 5% rabbit blood allows detection of the more fastidious actinomyces. Hecto and enteric agar. Hecto and enteric agar is a selective and differential medium used for the isolation and differentiation of enteric pathogens from contaminated clinical specimens. Fermenters such as E. coli produce colonies that are yellow-pink. Shigella spp is green or transparent, and Salmonella spp is green or transparent with black centers. Hemin-supplemented egg yolk agar, it is similar to neomycin egg yolk agar. Isolator or lysis centrifugation tube, Wampole Laboratories. The isolator is a unique system for the purpose of recovering organisms from the blood through a simultaneous process of lysis and centrifugation. The system is especially good for the recovery of dimorphic fungi, yeasts, mycobacteria, and Bartonella spp. Recovery of anaerobes, Haemophilus spp, and pneumococci may be reduced. Isosensitest agar and broth. Isosensitest agar and broth are media used for susceptibility testing in countries outside the United States. John E. Martin Biological Enrichment Chamber, GEMBEC, BBL, and Intray GC System Transport Medium, Biotest. The GEMBEC and Intray GC devices are transport or inoculation media for direct plating of specimens for the detection of N. gonorrhoeae kanamycin vancomycin lake sheep blood. Agar kanamycin vancomycin lake sheep blood. Agar is an enriched, selective, and differential medium used for the isolation and cultivation of anaerobic bacteria, especially slowly growing and fastidious anaerobes from clinical specimens, such as Bacteroides spp. And Prevotella spp. Lactobacillus emeris broth. Lactobacillus MRS, Deman, rugosa, and Sharp. Broth is a non-selective liquid medium used for the isolation and cultivation of lactobacilli from clinical specimens and dairy and food products. Leventhal agar with bacitracin and H. influenzae antiserum. It is similar to chocolate agar. Lemsime. Lothanai is a modification of Todd Hewitt broth and is an enriched selective liquid medium used for the isolation and cultivation of Streptococcus agalactiae. Lithium chloride phenylethanol moxilactam agar. Lithium chloride phenylethanol moxilactam agar is an enriched and selective agar used for the isolation and cultivation of Listeria monocytogenes. Loeffler's medium. Loeffler's medium is an enriched non-selective medium used for the cultivation of Corynebacteria, especially C. diphtheria. Lombard dowel egg yolk agar. It is similar to neomycin egg yolk agar. Lowenstein-Jensen medium. Lowenstein-Jensen medium is an enriched non-selective medium used for the isolation and cultivation of mycobacteria. It is similar to the American Trudeau Society medium in its content and its ability to grow mycobacteria. Lowenstein-Jensen medium. Gruft modification. The Gruft modification of the Lowenstein-Jensen medium is an enriched selective medium used for the isolation of mycobacteria. Penicillin and nalidixic acid are added to the medium and inhibit gram-positive and gram-negative organisms, respectively. RNA is added as a growth stimulant. Lowenstein-Jensen medium, Mycobacticell modification. The Mycobacticell modification of the Lowenstein-Jensen medium is an enriched selective medium for the isolation of mycobacteria. Cyclohexamide, lincomycin, and nalidixic acid inhibit saprophytic fungi, gram-positive organisms, and gram-negative organisms respectively. No RNA is added. Lowenstein-Jensen, medium with 1%, ferric ammonium citrate. Lowenstein-Jensen, medium with 1%, ferric ammonium citrate, is an enriched and selective egg-based medium used for the recovery of Mycobacterium haemophilum. Ferric ammonium citrate is the additive that allows this organism to grow. Lowenstein-Jensen medium with 5%. NaCl Lowenstein-Jensen medium with 5%. NaCl is an enriched selective medium used to differentiate sodium chloride-tolerant strains of Mycobacterium. Most rapid growers, e.g., the Mycobacterium fortuitum complex, as well as the more slowly growing organism, Mycobacterium trivial, will grow on this medium. Lysis centrifugation tube. It is similar to isolator. McConkey agar is a selective and differential medium used for the isolation of gram-negative organisms. The nutritive base includes a variety of peptones. The medium is made selective by the incorporation of bile, although at levels less than those used in other enteric media, and crystal violet, which inhibit gram-positive organisms, especially Enterococci and Staphylococci. McConkey agar with sorbitol, S-MAC. S-MAC is a selective and differential medium used for the isolation and differentiation of sorbitol-negative E. coli. McConkey broth. McConkey broth is a differential medium containing the indicator bromcresol purple used for the detection of coliform organisms from contaminated food, water, or stools. Mannitol egg yolk polymyxin B. Agar mannitol egg yolk polymyxin B. Agar is an enriched, selective, and differential medium used for the isolation of B. cereus from mixed clinical specimens. Mannitol salt agar. Mannitol salt agar is a selective and differential medium used for the isolation of S. aureus. Chaus. Martin Lewis agar. Martin Lewis agar is an enriched and selective medium for the isolation of N. gonorrhoeae. MB Bacti alert, Biomérieux. MB Bacti Alert contains a modified Middlebrook 7H9 medium, supplemented with casein, bovine serum albumin, and catalase. It is used with MB Bacti Alert 3D, Biomérieux, Inc., for the cultivation and detection of mycobacterial growth. MGIT, Mycobacteria Growth Indicator Tube, BD Diagnostic Systems. MGIT is a Middlebrook 7H9-based broth system that contains a fluorescence indicator, which is used for the detection of mycobacterial growth. Middlebrook 7H10, Agar Middlebrook 7H10. Agar is an enriched non-selective, agar-based medium, used for the isolation and cultivation of mycobacterial species. Middlebrook 7H11, Agar Middlebrook 7H11. Agar is a non-selective, agar-based medium, used for the isolation and cultivation of mycobacterium species. Middlebrook 7H9 broth with glycerol. Middlebrook 7H9 broth with glycerol.